Craig's a soil microbiome researcher at Flinders University in Adelaide, using DNA-based techniques to study links between biodiversity, soils, ecosystem restoration and human health. So those of you who have listened to Stacey's presentation or Dan's presentation or Felice's presentation, I think Craig will be building on a lot of that, and it should tie everything up in a neat bow, right, Craig? Please welcome Craig.
Thanks, Ellie, for that introduction, and thank you to Matthew and the organisers for the opportunity to speak to you today. My title here, Soil Ecology is Human Biology. It sounds a bit of a stretch, but what I'm hoping to convince you of is that the microbes that live in our soils and our environments provide a tangible biological connection between the health of environments and the health of people. So yeah, we'll start by acknowledging that human health has many factors, or human health and disease have many different factors that contribute to them, and soil ecology is part of that story. We've heard some excellent speakers talking about nutrition, and I'm not talking about, you'll have to sort of draw the lines here where nutrition comes into it, because they interlink quite a lot. I'm going to be focusing on some microbiome mediated pathways between environments and human health, which will hopefully become clearer as I talk. But yeah, lots of linkages with those other presentations. So my research, I guess I should say my team's research. These things are a team activity. Two different sort of overlapping parts here where we're studying the shifting of soil microbial communities with ecosystem condition. I guess I'm using language of ecosystem restoration, etc. The principles are generally applicable across production systems versus environmental restoration. So seeing how those communities change in different types of environments with different condition of environments, and also how those same types of organisms can be overlapping and potentially contributing to our health. Some terms I'll use, which you've heard today, the microbiome. What is that? It's a community of microorganisms living together in a particular habitat, in the gut, on our skin, in our airway, in the soil, in the roots, the root zone of a plant, the rhizosphere, in the ocean, etc., on leaves, inside the roots. They're different types of microbiomes. To get sort of a little bit more technical, we tend to use the word microbiota for the community, and then microbiome. The definitions will vary, but we tend to use microbiome for that community, plus the genetic material or the DNA, plus the sort of metabolites or breakdown products of those communities. So how's all this linked to human health? Well, funnily enough, we are mostly microbes. We like to think that we're a standalone organism, a human. We're really a system with pretty permeable boundaries. Life is systems all the way up and all the way down. And all of these, I guess almost all of these microbes come from the environment, specifically how you're born, whether or not you're breastfed, from your diet, and actually from the environment through the air we breathe. There's an aerobiome, soils and from plants. This stuff blows in the wind. Where sort of you hang around in an environment for half an hour, you're getting microbes from that environment land on your skin, get in your airway. It's getting into your lungs, and there's mechanisms that will push those microbes from your lungs to be swallowed into your gut. So, and we can think of microbiomes as our own sort of personal workforce, or army, if you like, or a toolbox, or even a personal psychologist. We heard a bit about that sort of the mind-gut microbiome link earlier today. And they're fundamental to our health. Every day it seems there's a new study talking about how microbiomes, often in the gut but also elsewhere, how they're linked to different diseases and also supporting health. I just wanted to acknowledge my particular research role. I'm an early career researcher. I spent sort of 15 to 20 years in state government in South Australia in the soils program. The 15 to 20 is sort of, there's a PhD sort of in there, and I finished that in 2019, so I'm an early career researcher. My postdoc position now is funded by the New Zealand government, or majority funded by the New Zealand government, in a people, cities and nature research program with 4 different sort of activities. And it's about putting nature back into cities. And one of those components is this idea of studying the restoration of health promoting soil biodiversity, which is what I'm focused on. We're partnering with the University of Waikato. So yeah, I'll dig into the details a bit about that first component, how microbes shift with changing ecosystem condition. And apologies to people who've sort of seen this stuff before. I know you guys do get some pretty good education and know a fair bit yourselves, but as a plant grows, in the background or bulk soil, there's a whole bunch of different microbes. Diversity is there. But if there's no plants growing, they're probably just hanging around, not doing much. So the biomass levels in the microbial communities is low, but there might be lots of diversity. As a plant grows, it'll push out those exudates and recruit certain members of those communities that do really well. And in and around the rhizosphere, I guess a selection will be favoured and sort of grow up more and have more biomass. So the graphic there is about the network complexity of a selection of microbes that are promoted by certain plants. Getting into what shapes soil microbial communities. I mean, you'd be familiar with what sort of different plants grow in different environments that you can see out there in the landscape, and it's kind of the same sort of principles, different sort of pH, organic carbon, oxidation status, or redox potential is kind of a concept that when you're talking about anaerobicity to aerobicity, moisture, and then sort of climatic factors, temperature, etc., and then to plants, the type of plant, the diversity of plants, all these things will shape the communities in the environments. And when things are deficient versus sort of more moderate to extreme, you'll get sort of conditions that favour different types of organisms or can be more limiting to organisms. And then you'll get sort of these plant-soil feedbacks where plants will promote certain members of the community. They will then provide services and benefits back to the plant, and you can get this soil conditioning, the conditioning of the physical and chemical structure of the soil over time due to those sort of two-way sort of relationship there. And I guess more plants, some of you guys will be very familiar with this stuff. More plant diversity, you have much more interactions in the sort of root zone connections. So this is an example from a paper where you've got a maize plant, it's pushing out exudates that favour formation of rhizobium in a legume that's next door, and then that's fixing nitrogen. Excess nitrogen will then go back to the maize plant. So this is kind of working better together principles. And more diversity means you've got much more diversity of habitat, more root zone interactions, so then more microbial community diversity below ground. And there's different types of environments in terms of, here's some terms, rhizosphere, detritosphere. I hadn't really heard of that before until this paper. This is, obviously one is around the root zone, one is sort of around stuff that's decomposing. You've got your bulk soil. This is another term I sort of just found, hyphosphere, around sort of fungal hyphae. There's also endosphere, which is inside the root zone. And we sort of heard about seed, core microbiomes in seeds as well. Moving on to, I guess, the relationship between microbes and nutrients. So if you've got too much facility in your soil, your plant's going to lose that incentive to invest in root production, to invest in the relationships with the root sort of fungal mycorrhizal relationships. So the two sort of sides to that picture just indicate excess fertility. It's really losing the incentive to grow the roots and grow relationships in the soil. And this kind of builds on what Dan was saying, that just the right hand panel here, we know a lot more conventionally about how inorganic, if we just sort of take the case study of nitrogen as a major nutrient for plants, I guess we know a lot more about inorganic forms of nitrogen, so nitrate and ammonium as being plant food, if you like. But recently, this is a paper from 2021, we're coming to understand that more organic forms of nitrogen are also used as plant food. So whole bacteria in this phenomena called rhizophagy, where plant roots will more or less eat bacteria, if you like, and use them as food, and then breaking down bits of bacteria to proteins and peptides and amino acids and that sort of thing. So a variety of organic forms of nitrogen are also plant food. Yeah, and if we want to think about inoculations or amendments with microbes, think of them as maybe an army to do work for you. Also think about, have they got the appropriate shelter and habitat? Are they being fed? What's the sort of existing force there that they've got to come up against? So you can't just sort of throw microbes out there and expect it to do magic. So here's a bit of an analogy, because soils are different. So on the left-hand side, we have the dirty old kitchen sponge, and that's sort of my metaphor for more organic matter and clay content. That's going to be better habitat for microbes versus a very sandy, coarse sandy soil. There's not many nooks and crannies for nutrients to hang on to or for microbes to hang on to. So they're sort of different habitat, if you like, for microbes, and your soil type plays a big role. These are some of the techniques we're using. So we take a soil sample, and the same techniques you can apply to fecal samples, if you like, stool samples for gut microbiome. So take a soil sample, we're extracting DNA. I don't do half of this stuff because I'm a computer person, but we have wet lab people that do a bunch of this steps in the pathway. So there's a step to extract DNA, and then there's a couple of different options. There's a bunch of different techniques, I suppose, to analyse soil biology. These are a couple that we focus on in our team. And essentially it's about asking who is there and what are they doing. So we're community profiling, if you like. The top sort of pathway, we're analysing a particular region of a gene that's reasonably good at identifying, say, bacteria or fungi to a certain level. It's a bit cheaper than the other method, and we can kind of ask who's there or assign taxonomy by extracting DNA, amplifying it, sequencing it, matching that up against databases to get an idea who's in that community. And it's kind of like a bird survey. You're counting different numbers of birds and giving them a count or a number. So we're counting different types of bacteria and giving like a relative abundance of those. The second approach is taking all the DNA, randomly chopping it up, sequencing it, again, matching it up against databases, and you can either ask who's there or kind of what are they doing, so more of a functional role, which is probably a more powerful approach. Costs more money. So just as a guide, sort of $100 a sample-ish, $60 to $100-ish, and then this has come down to about $350 a sample-ish. So using these techniques in post-mining scenarios in Western Australia, the photos are of what we call a restoration chronosequence. So they're different ages of vegetation post-mining. And so the plot here, through characterising the communities of bacteria and doing a measure of similarity to a target unmined reference native vegetation, we can sort of look at how long it takes for doing revegetation, rehabilitation from bare earth, basically, to try and build back something that resembles a native forest. And it can take 40 to 60 years for that community to sort of go back to a level. And then the native references will have their own variation, but to sort of get back to that level takes time. Now, moving on to, what's all this got to do with human health? Medical researchers and immunologists were basically seeing in the right panel plot here this sort of rapid rise in allergic, autoimmune and chronic inflammatory diseases over recent decades, and they're diseases of immune dysfunction largely. And at the same time, we've sort of seen a lot of people moving to cities. We've observed biodiversity loss, which is the indicators of there are global sort of biodiversity indices sort of going down. And just general reduction in our exposure to nature. And so these immunologists and medical researchers are asking the question, are these two things linked? And this is encapsulated in the idea of the biodiversity hypothesis. And soils are, if you like, the richest reservoir of microbial diversity in the environment. A recent study actually said that soils house something like 59% of all life on Earth. So they're really biodiverse. And essentially, is our disconnection between natural microbial diversity, chiefly from soils, is that they're asking, is this responsible for this rise in diseases of immune dysfunction? And also at the same time, lots of people living in cities. Our green spaces in cities are becoming very homogenised in terms of the soil microbiomes that are in those green spaces in cities. They're becoming very much the same across the world because I guess they're managed very much the same. Yeah, so we can ask, are we too clean? I'm glad it's pretty long way past lunch, because this is a pretty extreme sort of example here, where we've got this is a traditional African hunter who's prepared, I think it was a zebra, prepared his lunch, butchered the animal, and he's cleaning his hands with the microbial rich stomach contents. That's how he's sort of washing his hands there. And this is kind of so far away from our concept of hygiene, it's not funny, especially going back to COVID. But yeah, this is their hygiene, microbial rich contents to wash your hands of the blood, etc. So yeah, I'd like you to think about the fact that we've co-evolved with bacteria. Essentially, it's building immune fitness, if you like, and we've got diversity here, potentially countering the pathogenicity of what he's getting exposed to. A few other concepts here. So we heard a little bit from the other speakers about, if you were lucky enough to watch them, about how we're losing some key members of our gut microbial community. So I guess antibiotic treatments, big doses of antibiotics, health episodes, stress, lack of sleep, poor diet and lifestyle. I'm sort of picking on hamburgers here. Hamburgers can be healthy, I guess. But yeah, anything, poor diet and lifestyle, let's say that, can cause the loss of key members of our gut microbiome, and that's a problem. Another interesting factoid, I guess, is that our gut is dominated by spore-forming bacteria, and spores kind of sound bad, but it just means that it's anaerobic in here, it's aerobic out here, and the bacteria that are anaerobic, but they need to sort of hang out in the environment, sometimes they'll form spores to just hang around until they can become active again. So our gut is dominated by these spore-forming bacteria that come and go. And maybe they have a home in the environment, which we'll get to. Let's just check our attitudes to microbes. They're bad, right? Microbes are bad, germs. So this is kind of how we think in hospitals. This is like a conceptual dose-response curve, if you like. We've got good health high on the y-axis, poor health on the low on the y-axis, and then I guess exposure to any particular microbe, E. coli, salmonella, etc. on the x-axis. And this is kind of how we think in hospitals, because we've got sick people, immune-compromised people. But should we think like this everywhere? Perhaps not. And the sort of thinking around the biodiversity hypothesis, which is about exposure to natural microbial diversity builds immunity, and a related theory called the old friends mechanism, which is that some key species may perform critical roles for us. So build those two together, and instead of having a straight line, maybe we should be thinking about this U-shaped dose-response curve. And this kind of aligns with this sort of hormesis or principle, that a little bit of something is potentially good for us. If we've got and you'll kind of recognize this from plant nutrition. When you've got a deficiency, it's obviously bad. If you've got sufficiency of nutrients, that's kind of where the magic is, and then things can get toxic if you've got too much of anything in particular. And this kind of U-shaped dose-response curve is kind of generalizable across the whole field of biology, really. But so where that sort of Goldilocks zone is, or the sort of moderate exposure to microbes, at the same time they're potentially the community, they're keeping control of pathogens through that diversity, they're keeping control of each other. So instead of a hospital sort of thinking, this is possibly something that we need to move to. And this is kind of the idea of it's not called the hygiene hypothesis anymore, but the biodiversity hypothesis. And a bit of dirt never hurt, but it's kind of got to be the right sort of dirt. So so we lose microbes from our body. Potentially, where does that get replenished from? From the environment. So this is the idea of two layers of protective biodiversity from the environment and within ourselves. All right, bit of detail here. So the picture here is of the cells of the gut lining. So I'm going to tell you about 4 different ways that microbes in our gut and coming from the environment can benefit our health. So they can sort of add to protective communities that line our gut. So they're either holding ground or starving the pathogens of nutrients. So protective commensals. Commensals kind of just means harmless, but potentially they're beneficial. Immune signaling. This is really interesting. So the particular bacteria, the molecular patterns in them, They are, the diagram there shows a dendritic cell of the immune system. It's reaching through the cells, it's sampling a bacteria, and it's going, hang on, yeah, I recognise you, you're a friendly or you're a foe. So these things are participating in the immune signalling, and they're either triggering defence or they're triggering tolerance. Most people know about triggering defence, like inflammation, but triggering tolerance is a thing. They can build immune memory through antibodies. And then lastly, there's probably more mechanisms, but this is kind of 4 that we know about, the beneficial functions and sort of breakdown products. And these are the kind of compounds, or starting compounds and then producing other compounds that Stacey was talking about. And this kind of looks a bit busy, but we've just got like 2 scenarios here where, from an evolutionary point of view, we were sort of, when we sort of evolved, exposed to sort of a lot more natural microbial diversity, we had a sort of a balanced microbiome from that. And then with land clearance, monocultures, sort of degraded pollution, etc., we're being exposed to different sort of environments, different communities. We've got potentially a microbiome that's a bit out of whack. All right, so where's the evidence? On the left, 2 studies here. On the left, they're saying that kids that grow up on farms have lower rates of asthma and allergy. On the right, they're saying, okay, within farm kids that are exposed to more traditional farming practices, so a bit more exposure to soil and manure, they have even lower allergic and asthma incidences. This was a study from a group in Finland, taking sort of the concrete sort of daycare yards compared to a treatment. They brought in soil and plant materials, and within a month the kids there had, there's a number of studies associated with this, I'm just showing one, but they showed changes to gut microbiome and skin microbiome and benefits to their immune biomarkers for expressing a more tolerant immune responses. So just exposing the kids to soil and plant material, so being around nature. Yeah, and I guess you do mouse studies when you really want to control a whole bunch of things. So mouse studies where they're bedding them with soils that is changing their microbiome and building these immune biomarkers. A few other different ones here. Accelerating recovery from antibiotic treatment just due to sawdust exposure. Housing mice in a farm yard type habitat or bedding protects them against colorectal cancer. You might have heard of this Mycobacterium vaccae, more or less an antidepressant sort of effect from that particular soil-associated bacteria. And from Felice's talk this morning, I think it was you mentioned that some of these pathways are through the immune system, and this one was shown to be through the immune system that this particular effect was happening. Yeah, so not all soils are the same. This is a bit of a disclaimer. So, and they can vary just naturally. Soils are going to vary, but also in any given soil type, they can be in poor or sort of good condition. And I guess you guys know what poor versus good condition is as to how the plants grow and how the animals are, the health of the animals. So yeah, these principles are, I guess, applicable across animals, and plants have immune systems too. So microbes are part of this story. Just a couple other quick ones here. Antimicrobial resistance we've heard of, and pathogens, they spend time, potentially survive and hang out in the environment, but microbial diversity in the soil can act as a barrier and help suppress those bad guys. Some evidence or some studies I did in my PhD, I was interested to know if these ideas were there in some of the evidence from Australian landscapes. We didn't have a measure of soil microbial diversity for Australia. However, I knew a bit about soil mapping, and we did have a map of soil cation exchange capacity. So does anyone know what that is? Yeah, so sort of organic carbon and clay content kind of bunched together. Cation exchange is about, just more or less fertility related to organic carbon and clay content. And I brought in some other data sets that had microbial data and was able to sort of show that this layer of soil cation exchange capacity was a good enough proxy indicator of diversity in diversity, just meaning I guess, the number of different types of organisms for bacteria and fungi and multicellular animals. So didn't have microbial diversity, but had soil cation exchange capacity as a proxy, and then took some local government area. This is a plot here. On the y-axis is hospitalisation rates for infectious and parasitic diseases on the y-axis. So higher there is worse outcomes for infections, I guess, and hospitalisations. And if we just sort of focus on the ring, on the circled area there, this is in a lower socioeconomic group where you kind of tend to see environmental influences show out in human health outcomes. So in local government areas that were in and around lower soil cation exchange capacity or typically lower microbial diversity, they had much higher rates of infectious and parasitic diseases. So it's kind of a lower quality type of evidence, but it's pointing in the same direction, that less immune fitness where you haven't got that sort of environmental effect. This is another study I did, and the little graphic there talks about sort of more or less a monoculture. It was kind of wasn't a pasture, but it was kind of a degraded pasture-like thing that was used as a fire break where we were sampling. So pretty crappy degraded land through a restoration chronosequence or a number of different sort of samples with increasing vegetation, above ground vegetation, and was able to show, well, I was interested to know what type of bacteria were trending at that end and towards that end. And kind of the simple message there was that if you have simple and unreliable food in your soils, it favours fast-growing generalists, the opportunists, kind of the weedy ones with potentially more pathogenic character at the sort of disturbed sort of side. And then at the more mature, above-ground established vegetation, they were favouring bacteria that were longer-lived, more niche adapted, and kind of working together to break down more complex foods. Then we thought no one will take us seriously unless we do a mouse study, because we're trying to build a bridge from environment to health. So we did a mouse study. This was kind of a bit risky. Some of my PhD supervisors didn't really want me to do this, but we did it. Had 3 different treatments. Lab mice in a little open top wire cage. It was kind of old school metal mouse racks, if anyone does mouse studies. We wrapped them in plastic, gave them a little vent so they could breathe, and then we had a tray of dust with a little USB fan for 7 weeks, sort of blowing dust over these things. And so it was other studies, they put soil in the cage or they inject stuff in their mouth, and we were sort of interested to know, trace level, sort of typical normal exposures. Is this going to make a difference? We weren't sure. So we had no soil, we had low biodiversity at a microbial level and coming from low above ground diversity, and similarly high diversity micro and high diversity above ground, these 3 treatments. And what we found was a shift in the overall sort of community composition of the gut microbiome of the mice. We did a anxiety-like testing where you video track mice in an open top box. Time spent in the corners or sides is sort of more anxious behaviour. More time spent in the middle is less anxious behaviour. And so there was it was interesting. There was a sex difference. So the females were actually more responsive to the sort of environmental treatment, if you like. But there was a particular microbe among all the different bacteria that shifted in the gut. A lot of them were just mouse intestinal bacteria, and I wasn't too excited. And there was one that I found that had been first characterised in a soil. It was spore forming, it was anaerobic, and it was butyrate producing. So, and butyrate is just a breakdown product of organic matter through fermentation. It's actually a key ingredient for the cells of your gut. It's kind of like their main food. So this particular bacteria had more or less has a home in soil. It's there in all of the guts, but it's more favoured in the high biodiversity treatment, and it correlated with the anxiety-like reduction results. So it kind of opened up a bit of more questions, and we had a PhD sort of student come along. So yeah, the idea there is that maybe there's bacteria. We sort of found maybe one of them, maybe there's others. where they have a home in the gut, maybe they have a home in anaerobic, occasionally wet, organic rich soils, and they form spores when they dry out and sort of blow around, and they come in and they go and they... So yeah, potentially there's this sort of idea of overlap. And the spore forming allows them to, I guess, hang around and persist and survive in between when they become active. So yeah, we had a PhD student, Joel, who took that further and really focused on this idea of butyrate-producing bacteria. And he characterised sort of globally where they were out in the environments and different types of environments. Yeah, so I guess I'm a bit of a computer nerd. I like to analyse the data and sort of see where it can go and look for patterns. So I've been spending time more on that functional side of it, and I guess this is sort of concept of functions are sort of overall pathways of high-level sort of descriptions of what's going on, and then you have reactions, and a reaction is like acid plus base equals salt plus water, or we saw a reaction about sort of the carbohydrates being formed from sunlight and other reactions. And then compounds is all what Stacey's talking about, this sort of array of different chemical compounds that are out there. So I'm interested to know what is the functional role of these communities. And this paper here looked at in different soil environments, in different types of ecosystem condition, but also in human health and disease from gut microbiome samples. So yeah, and I guess you can see here a little graphic of a reaction where you've got a few molecules, glucose here, you've got an enzyme or a catalyst, and then you've got a few sort of breakdown products. So the compound level is kind of at the detailed end, and the function is sort of summarising that overall. And the standard way, the tools that are available, the standard way that this information is presented is kind of as this functional pathway sort of level. So this is kind of a bit of a diagram that talks about a whole bunch of stuff happening, and there's a bit of red stuff highlighted, and that talks about the functional pathway. And I sort of struggled with that concept a bit because I'm not a biochemist, and it kind of I couldn't really understand or try to draw patterns from that, because you get 10 to 20,000 sort of functional pathways, right? In one sample, that sort of adds up to 100%. And if you've got 20,000 different functional pathways, what does that all mean? Like, it's a bit hard for me to get my head around. So I wanted to flip that around to look at, okay, what is that 100% per sample? Where is that sort of allocated across different compounds? So that could be compounds of sort of vitamins and the phytochemicals and all those sort of different things that may or may not be related to health. So sort of turning that around a bit through some computer magic. Yeah, because I think we know more about compounds, chemical compounds, molecules, vitamins, etc. We've done a lot more work on that in terms of health links. Compounds are more or less, substrates or feedstocks that are in our diet, in the environment. We're interested in organic carbon. That's, made of compounds. So they're often things that are desirable or undesirable. There's a lot of focus on compounds as opposed to functional pathways. So I wanted to flip the whole thing upside down to understand where the genetic sort of weight of activity from the DNA of these microbial communities, where it was being allocated across these different compounds. That's kind of a bit complicated. What I was doing, I was mapping it into this space, pretty simplistically mapping it into this space of, this is kind of a compound mapping system that was used by fossil fuel resource assessment. So you've got sort of oxidation reduction here. It's basically based on the chemical formulas of all these compounds. So oxygen to carbon ratio, hydrogen to carbon ratio on the y-axis, oxygen to carbon ratio on the bottom. So kind of coal is down here, and woody biomass is here, and jet fuel is up here. So, but you can map sort of big chunky compounds on here, but you can also map vitamins and a whole bunch of other biomolecules onto this sort of, it's fairly simplistic. I'm only considering oxygen, carbon, and hydrogen. So the next step will be to bring in nitrogen as well. So I'm mapping on here. This is where generally the human gut microbiome sort of is just distributed across. If this is a map, this is sort of like mountains and valleys and peaks and depressions of activity. So lots of activity around carbs, much less, but still a fair bit in fats. Kind of the main categories: fats, proteins, amino sugars, carbs, tannins, lignin, condensed aromatics. And this is kind of where, in soils, it was a very similar distribution or weight of sort of activity or focus on the sort of spread of compounds. And this is kind of quite simplified, big sort of coarse, chunky categories. And I also sort of looked at the weight of activity in and around particular biomolecules. And so this is just visually where all the sort of the activity or weight of sort of genetic capacity is happening. They're all human gut samples, they're all soil samples. So they kind of look like there's a fair bit of similarity there. They're a bit different, but they're in the same sort of area. Ooh, this one's a little bit hard to explain, but so I've got lipids, proteins, amino sugars, carbohydrates, condensed aromatics, lignin, tannin, and instead of like lots of dots, I'm taking a weighted average to sort of give it a coordinate in every sort of sample. And the colours are blue is young, yellow is old or unmined reference. And so this is showing how, from a microbial community point of view, embedded in the DNA, it's saying older, established, mature, ecosystems, their soil microbiomes shift because, you see it in the environment. There's different things on the ground. Those microbes are tailoring and gearing up to produce those different things that are there to eat. And you see it here from their DNA. That's just kind of an overview results. I looked at heart disease, colorectal cancer, type 2 diabetes, and problem behaviours, and sort of with broad classes of compounds and in and around specific sort of vitamins. A little bit reductionist, but I was trying to go community as well. But yeah, strong signals in heart disease and colorectal cancer, less strong in type 2 diabetes and problem behaviours. Had to dig into a sort of finer resolution for those. But this was this is kind of a network plot for type 2 diabetes, where it's about what are the significant correlations of compound activity, and sort of on this plot here on the right, we've got normal, healthy, and then this is untreated type 2 diabetes here. So there's just far different relationships in terms of what compounds are being processed in this disease. And type 2 diabetes is apparently quite hard to diagnose, quite complex. Definitely something happening there. So this is kind of where I want to go with now. Instead of just 2 dimensions, I'm bringing nitrogen into it as well. This is a data set post-mining in the US, and this is kind of a synthesis or results plot where from, say, 5 different age groups, I'm saying what is increasing or decreasing as you go from really young to really old. And so green dots are increasing, red dots are decreasing, but with age. So imagine this in a disease case. The red and the green, red versus the green, are potentially, different mechanisms of activity, potentially their targets for intervention. So the same types of analyses we can look at what's happening in the environment and what's happening in human health, given the sort of overlaps. But this is sort of where I want to go next. But conclusion, soil and ecosystem quality, affects microbiomes and their capacity to process compounds. Or it's like, I was speaking to Dave this morning, and you guys will be familiar with minerals, applying minerals if they're deficient. So this is kind of about making the bioavailability of those minerals more or less because of the activity of the microbes. Substantial overlap in the gut and the soil. Perhaps at least some of these are shared, maybe many of the microbes themselves. And just this idea that hanging around in different environments may actually be weighting or biasing, shifting our gut's functional capacity to process foods, and that could be affecting our health. So it's kind of like another layer to the nutrition and the diet, just the fact that the engine room, the sort of workhorse, that the middleman between the diet and our health is kind of the microbiome. And if we're shifting the capacity, the strengths and weaknesses of that microbiome, that's potentially influencing our health. So, that's it.
Well, that was amazing. Thank you so much, Craig. You describe yourself as just a humble early career researcher, data nerd, computer nerd. But I'm looking forward to seeing what the late career Craig has produced. And Yes. But yeah, what I took away from that, some incredible insights about the ability for exposure to the microbiome in soil to reduce allergy, to reduce disease, to increase immunity, to reduce depression and anxiety. Pretty amazing. So thank God for computer nerds for doing all of that work to understand what's actually going on there. Thank you. So now we have plenty of time for questions, 15 minutes or so for questions. And I'll remind you, Craig, to repeat the questions into the microphone. Over here, maybe we'll start over here.
Just a technical question first. When you looked at the correlation between the compound processing potential and the genetic resources, and it looked like it kind of looked similar between soil microbiomes and gut microbiomes. Did you look at it any just like a random microbiome from somewhere else, just as a comparison, to just see that that's not just how genetic resources are compartmentalized in a microbial system?
Yeah, so the question was, when looking at how the genetic resources are allocated across soil microbiome versus gut microbiome, did I look at other sample types to see if that just wasn't a general thing? No, I didn't. And it's a good question, but yeah, it's worth looking into, yeah. Yep. Okay. Well,
There you go, Craig.
okay.
Thanks, Craig, for your talk. You mentioned the mine restoration, and you said, I think it was 40 to 60 years before you got a healthy microbiome back in the soil.
Yep.
Could you accelerate that by planting diverse vegetation or more diverse vegetation and inoculating with the microbiome?
Yeah, you definitely want to try and increase that speed. Yeah, so this was kind of data that came from existing work that had sort of been done previously, and that I showed you one way of analysing it, and there's a few sort of slightly different ways to analyse it. You get similar results, but in some of the plots, and what they actually do is direct transfer of soil. So a place they're going to mine, they will take the soil out of that in 2 layers and they'll put it into their sort of old mine site. So they are kind of inoculating, I guess, bringing in soil over that's appropriate. And even in slightly different ways of measuring it, you see a bit of a, when you're measuring similarity to the reference, you see a bit of that inertia, biological inertia, if you like, and then it goes down and then it comes back up over time. So you kind of need, there's facilitators and followers, if you like, there's across the community. So yeah, some of them probably definitely need the living healthy plants and a mixture of plants, and they did plant a biodiverse mixture of plants. So they're trying to do their best, even though these restoration efforts have improved over time or changed over time, but they do try and do that. But maybe there's other ways to accelerate it that we need to look at. So, yeah. Yep.
I was wondering if you looked at the ratio between, in like a microbiome, the ratio between fungi and bacteria, because I know in soil health, that's like often a test.
Yep.
Like a more healthy soil, I think, has more fungi than it does bacteria, and I was wondering if you looked at that in a gut microbiome.
Yeah, good question. And there are different techniques. So did we look at the ratio of fungi to bacteria in our measures of the microbiomes? No, we didn't. Just that the particular techniques that we're using, with the first one, the amplicon or marker gene stuff, it's pretty much dedicated to bacteria or dedicated to fungi. You can't really sort of bring them together. They're like a relative abundance out of 100%. Just in bacteria or just in fungi, so we couldn't do that. In shotgun metagenomics, I guess in theory we should be able to do that, but just the sort of physical sampling and the processing, the wet lab procedures, they probably are more favoured to bacteria, so we couldn't. There's other techniques that you can do that. We didn't do that. Yep.
Sorry, just a very quick follow-up question to that. Do you know about any sort of recent advancements in the bioinformatics space to replace that wet lab process with something more automated, so you could get models of those things together?
There seems to be oh god, hopeless.
You're not the only one, Craig. Everyone. It's some processing thing.
Yeah. Have there been advances in bioinformatics or tools to help move straight into maybe the fungal bacteria ratio calculations? Yeah, I think Felice mentioned there's lots of money thrown at this space from probably a human health point of view. There's lots of tools and hardware and software bioinformatics sort of pipelines being developed around the world. I'm not aware. I probably can't keep up with everything because there's a lot happening. I'm not aware of that. So yeah, I guess they're. Yeah, I don't know. I don't. They all have pros and cons. All the different tools and techniques and pipelines all have pros and cons. So you can't sort of do everything with one technique. Back.
I'm curious to know, is there, of all the biome, what proportion has a life cycle that is outside of us? You know, is it just a matter of feeding them correctly, or do we actually need to be reseeding our own biome?
So the question was, do we? Can you say it again? Do we? Yeah. Go again.
Again, I don't know if this falls outside of your realm of expertise, but yeah. So, like, do we need to be eating living organisms to be maintaining our own biome, or do we just feed them prebiotics?
Yeah.
You know, if we do that well enough.
Cool. So do you need to be eating live organisms or just eating properly to maintain our microbiome? So you definitely lose key members. Where do they come from? I don't know that you need... Well, probiotics can be useful. Yeah, I've certainly used probiotics at times, but the other sort of concept is that just being in and around different spaces... the aerobiome, you know, is one way. It's probably like a low efficiency way to get exposure. Things will work their way into your gut just through breathing because there's like this mucociliary escalator thing. I don't know if I've got that term right, but essentially from your airway, it'll work its way to the top of your got a lot here where I'm not an anatomy person, but yeah, essentially it'll work its way into your gut just from breathing. Probably, you know, digging in the soil, those sort of are probably higher exposure levels to sort of get that. Being in and around different types of nature, I think will get some exposure. Eating perhaps the odd bit of raw food, maybe not washing your hands so much after you pat the dog. I don't know, sort of bordering on. I'm not a doctor, so I'm no health advice, but yeah, obviously hygiene with raw chicken. But I don't know if that answered you. Did that answer your question?
Wash your hands with the stomach contents of...
Yeah, I mean, we've probably, in the past, we definitely got live organisms through probably our food, through our diet directly.
It's just that it was curious to me, and I'm a complete very ignorant. It was curious to me that the actual life cycle is outside of us at times.
Yeah, and I think that's a really interesting idea that potentially things that have a home in us sometimes also have a home in nature. Oh, that's pretty interesting. Yeah, and I think that's true.
Just, can I follow up on that question? I was just wondering, how do a lot of these microbes end up in our gut? Because they have to transit through the stomach, right, which is pretty low pH.
Yep.
Is it spores that get through, or is it just like a flood the zone approach that, and then some of them make it through? You know?
This is bordering on, you know, I'm a computer guy, but I'd say spores. Yeah, I think even with the techniques that we use to characterise these communities, they have trouble breaking the spores and actually identifying them. That's a thing they've had to work on. So yeah, spores would be definitely a way they can pass through the different pH levels and different chemistries and maybe it's like certain triggers of redox potential and pH and whatever that they then activate. But maybe they're encased in food as well. Maybe. Yeah, I'm not sure. Good question.
Question, Craig.
Yes.
You were talking about the analytical tools to work out who is there in the soil and what are they doing. Just thinking about Stacey's talk about nutritional dark matter, and I'm thinking about soil microbiome dark matter. How much do we actually know? Like when you do that, how much of what you find do we know anything about?
Repeat your question, Craig.
Ooh. What's the extent of microbiome dark matter in the soil? Okay, there's two different sort of steps to that. Say with the shotgun metagenomics, we'll get gigabytes, hundreds of gigabytes of data with raw DNA sequences, and potentially only 15 to 20% of that makes it through to the next layer. So there's a lot of stuff that gets chucked out, data that is not kind of used, and then we're moving to the next step of characterizing things against reference databases, and sort of fungi is much less well characterized than bacteria, and even soils versus human gut, the soils are a lot less well characterized than human gut. So yeah, we have like probably in fungi, I'd say that the majority of sequences that you think are fungi are sort of unclassified unless at a really high level. So yeah, there's a bunch of unknowns.
Yeah, I'm just thinking like, how many billions of dollars do we need to be poured into soil microbiome research? And we can put that in the video.
So much, yep, billions, trillions, yeah.
We need the soil dark matter CRC
Yep.
with hundreds of millions of dollars.
That sounds good, yep.
I'm sitting here probably oversimplifying it, but we're looking at all your research. The biome's great in the soil. How do we actually get it? Apart from encouraging the kids to play with dogs, muscle battle, going against our regen farming, disturbing the soil, making dust on it. I'm oversimplifying, of course. But how do you actually? How do you get like when we were growing up as kids? You played in the dirt. You actually got dirty hands.
Yep. I think it's the occasional how do you, without disturbing soils too much, how do you get decent exposure to the soil microbiome? So I think it's having occasional disturbance or occasional exposure, getting in there with your hands, or every now and then. Veggie garden, yeah. Don't wash it all the time. Don't bleach your veggies, yeah. Just occasional disturbance, occasionally getting in there and getting your hands dirty. Time for a beer? Yeah.
Ingrid's got one.
So with this, is that people are doing fecal transplants now to try and help people with their gut microbes. Effectively, perhaps in the future, they could just sniff a bit of dust. Well, that study.
Was that a question? Yeah. No.
Yeah, no, I'm just wondering, could this be where it goes?
Yeah. So the question was.
That study in Sweden, where in a month things change, and that's pretty amazing.
Yeah, so are there alternatives to, I guess, directly targeting the gut through just exposure to soil, sniffing soil, etc.? So I think more studies need to be done to confirm that, but yeah, that's pointing that way. That and it's kind of like prevention versus cure. If there's something really badly going wrong, then you'd potentially do the sort of fecal microbiome transplant, and we've sort of talked to medical people in Adelaide that are doing that, and they've saved lives in really drastic cases where that's happening. So yeah, it's probably on that spectrum from preventative sort of exposures through to treating serious disease that you'd do.
Forest bathing, they could do forest tours.
Yeah, I think I've done some forest bathing, and I really like the fact that I didn't have to think about what was going on. But yeah, maybe there's a bit more soil needed in that.